and especially in this particular building. The first class I had as a student, in RIT in my 4 years here, was in this building just a few rooms away. And now here I am 16 years later, giving a presentation on my research at the Summer Math Institute. It's kind of funny how life has a way of bringing you back and kind of going full circle. So today I'm going to share some of my collaborative and methodological research in the area of immunomethylomics. In a strange way, this work actually has its roots here. The first, my introduction to research was here at RIT, and the project that I worked on dealt with mathematical techniques for blind source separation. So in other words, the separation of signal mixtures, like images or audio, into their components. And so while today's talk doesn't cover separation of audio or images into their sources, it will cover techniques for separating cell mixtures into their components and the relationship between that and cancer research. Second, the motivation for pursuing a doctoral degree in biostatistics stemmed from a co-op that I did while I was a student here at RIT. So knowing that this audience has a very diverse academic background, I've attempted to structure this talk so as to provide enough of the background. There'll be quite a bit of biological terminology, and so I'll spend kind of the first half of this talk kind of going over some of those terminologies to help make clear some of the methods and techniques that are discussed in the second half. So perhaps some people in the audience have seen this figure or something similar to it. The idea being that a single measurement only provides partial information, and sometimes misleading information at that, and that only by integrating multiple different measurements taken at different perspectives, i.e. data integration, can the scientists in this cartoon stand any chance at deducing that the thing in front of them is an elephant. So data integration is something that we talk a lot about in the academic medical center that I work at, and it's really more than just a buzzword. It truly is fundamental to obtaining a holistic understanding of some system or process. Now in the area that I work in, we generally think of data integration in terms of combining multiple different omic data types for understanding some disease or process. So integrating gene expression data, or genotypic data, or DNA methylation data. Another way to think about data integration, and that's really a central theme in this talk, is integrating the same data type, i.e. gene expression or DNA methylation, but measured in multiple different tissues throughout the body. So some of the early studies of epigenetics in cancer primarily focused on looking at patterns of expression or DNA methylation in the tissue itself, so looking at the tumor tissue. But more and more recently, groups are beginning to understand that looking in nearby tissues is really fundamental for understanding how disease develops and how it progresses. So this brings us to the esoteric word that appears in the title of my talk, immunomethylomics. So what we're really talking about here is we're talking about changes in DNA methylation, and I'll define that in a few slides, changes in DNA methylation in immune cells and the relationship to human health and disease. So I have two simple objectives for this talk. First is I want to tell you a little bit about the history of immunomethylomics and cancer with a focus on some of the analytical methods my team and I have developed to facilitate its study. So in order to appreciate and understand these methodologies, it's pretty critical that we establish a common lexicon. So we're going to go over a little bit of biology. We'll talk about what epigenetics is, I already used that term. We'll talk about what DNA methylation is and how it's measured. I then want to finish by describing how some of the analytical tools we've developed are being used to facilitate the study of cancer. So the story of immunomethylomics begins with DNA methylation. So DNA methylation is one of several different epigenetic mechanisms, and epigenetics refers to changes in the functioning of a gene that are not due to changes in the underlying DNA sequence. Epigenetic mechanisms are stable, but they are potentially reversible, and they're passed on through cell division. What's really important is that epigenetic mechanisms have the potential to control gene expression, and that does everything inside of cells. It's now widely accepted that epigenetic alterations in target tissues are causal to human malignancies. So to reconcile or to describe the relationship between genetics and epigenetics, I think an analogy is helpful. So a standard piece of sheet music is comprised of musical notes along with cues to the musician to inform them how such notes should be played. So the intonation, how softly or loudly should the notes be played. So that piece of music, even though the musical notes are identical, can sound completely different if played by two different musicians. So the musical notes are to cues as genetics are to epigenetics. So much in the same way that the same piece of music can sound completely different if played by two different musicians, cells with exactly the same DNA sequence and originating from a single cell can elicit a myriad of different phenotypes, as evidenced by the roughly 200 cells that exist in the human body. The epigenome encodes information that defines the function of cells, tells those cells what to do, and these cells remember when they divide. It's why a brain cell is different than a liver cell, which is different than a skin cell. They all share the same DNA sequence, but something, the epigenome, is turning on and off genes that define the function of these cells. So the cell specificity of the epigenome is really a key feature of the analytical methods that I'll be describing later in this presentation. So, like somatic mutations, DNA methylation alterations accrue with age and are believed to mediate the effects of our environment on disease incidence and to contribute to disease progression and treatment resistance. The fact that DNA methylation sits at the interface, the crossroads of our environment and our health, is really the driving factor explaining the exponential growth in studies of DNA methylation in human health and disease over the past 2 decades. So, what is DNA methylation? Put simply, DNA methylation is the process by which methyl groups are added to the DNA. So if we remember back to biology, DNA is comprised of 4 nucleotide bases, right? A, T, C's, and G's. DNA methylation occurs almost exclusively in the context of a CpG dinucleotide. And what that means is that's a C followed by a G in the DNA sequence. Now, these CpG sites are generally underrepresented throughout the genome. They're less prevalent than you'd expect by random chance, but they tend to cluster in these regions referred to as CpG islands. And those regions are often located near what's called the promoter region of a gene. Now, about 70% of human genes initiate transcription in the promoter. And what can happen with DNA methylation is it can actually prevent a gene from becoming expressed. If we methylate the promoter region of a gene, that gene doesn't express itself. And the reason that's important for cancer development, if that's a gene that is like a tumor suppressor gene or some gene that's critical for what that cell is supposed to do, that's a bad thing. We don't want to methylate that gene. So shown here is kind of a simple illustration, well, maybe not super simple, but an illustration of what I was just describing. So the lollipops in this figure, so let's see if I can get my cursor over here. The lollipops in this figure represent CpG sites, okay? This arrow represents the transcription start site, and in the top, we have a gene that is unmethylated. So these lollipops are not colored in, and that is admissible to the expression of this gene. On the bottom panel, we have that those CpGs are methylated. And what that can do is recruit enzymes that block the transcription of that gene. And here's why this is important. If we go back to high school biology or college biology, we learned about the central dogma of biology. And what the central dogma is we have DNA in our bodies. That DNA is transcribed into RNA. The RNA is then translated into proteins. Proteins do everything in the body. So if we disrupt this process by aberrantly methylating a gene like a tumor suppressor gene, then we disrupt the ability to create the protein that that gene actually codes for. And that can set the stages for disease development. So we now know what DNA methylation is and why we care about it. I'm just going to say a few things about how it's assessed. There's a lot of different technologies. Probably some of these technologies exist here on this campus. A lot of different technologies for assessing DNA methylation. So shown here is a plot that depicts some of these technologies, and it shows the average price per sample to run this technology as a function of the coverage. And what we're talking about here in terms of coverage is the number of CpG sites in the genome that technique is capable of profiling. So there's about 30 million CpG sites in the human genome, and whole genome bisulfite sequencing allows us to basically profile all of those CpGs to see if they're methylated or not in a given cell. But that comes at a huge cost. at around $5,000 per sample. So this technique is cost prohibitive for an epidemiological study of cancer and DNA methylation. The EPIC array strikes a nice balance between cost, coverage, and throughput, and is pretty much the standard technique for studies in which we're looking for aberrant methylation in the context of cancer. So a lot of the studies that I work on with collaborators involve using this EPIC technique for assessing methylation. So the way that this works is the methylation level of each CpG is quantified by taking the ratio of the methylated signal intensity for that specific CpG and dividing it by the methylated plus the unmethylated signal intensity. And that gives rise to what we refer to as a beta value. The beta value is bounded below by 0 and above by 1. The reason we call it a beta value relates to a statistical distribution called the beta distribution, the support of which is from 0 to 1. Now, when we profile DNA methylation in a biological sample that we've collected from someone, be it a sample of blood or a piece of tumor tissue, that tissue sample is comprised of millions of cells. Now, within a single cell, a given CpG is either methylated or it's not. It's a binary process. But because we profile methylation over millions of cells, the beta value can be loosely interpreted as the fraction of methylated cells at that specific position. To help you visualize what data from these arrays actually look like, here we have 22 chromosomes. Now each one of these lollipops represents a CpG, and those CpGs are somewhere on the chromosome. The color of each one of these CpGs reflect its methylation status as measured by the EPIC technology that I was just describing. So yellow is indicative of an unmethylated CpG, and blue is indicative of a methylated CpG, and black is kind of somewhere in the middle. So this is what the data actually look like that we obtain. Now this blue-yellow color scale is something that I'll use throughout the remainder of this presentation. Just know that yellow is unmethylated and blue is methylated. Black is somewhere in the middle. So for the more numerically inclined, this is what the data would actually look like in tabular format. So what I'm showing here is an excerpt of data from a project that I actually worked on. And this project involved comparing DNA methylation between tumor tissue and adjacent normal tissue that were taken from cancer patients. So each row of this table represents a different subject or a different person. We have 5 tumor samples, and we also have 5 adjacent normal samples. The columns of this table represent different CpG sites throughout the genome. So this is a label, just a unique code for a specific CpG that sits somewhere in the genome. And same thing with this. Now the actual data set, I'm just providing an excerpt here. These arrays profile around 850,000 CpGs. So I'm not going to list a table with 850,000, but suffice it to say this would go out to 850,000. So our data is an n by j matrix of these beta values. Now, typically what we want to do, if I'm working with a collaborator, is we want to look through all of these CpGs and we want to identify the CpGs that exhibit differential methylation or a different pattern of methylation between the tumor and the normal samples. Why would we want to do that? Because that can begin to give us insight into how the disease develops. If we identify a CpG that exhibits a different pattern of methylation, like this first one, which is lower methylation in the tumor samples compared to the normal, we then look in the genome and we ask the question, where is the CpG located? What gene is it located in? And what is that gene responsible for doing? If that gene is a tumor suppressor gene or an oncogene or has some other job that's critical for the cell, then that would be important information to know and can give us insight into how the disease is developing. All right, so this stage of the game kind of developed some of the lexicon and terminologies that we're going to use throughout. Now I want to take you back to 2009. So up to around 2009, a lot of the studies, cancer studies, looking at patterns of DNA methylation in cancer were really focused on measuring DNA methylation in the tumor tissue itself and comparing it to adjacent normal tissue. And around 2009, there was this growing interest in profiling DNA methylation in blood and comparing that between cancer cases and cancer-free controls. The use of blood for interrogating methylation, there was really 2 main reasons for it. For one, blood is a lot easier to get from someone than tumor tissue, right? It can be relatively non-invasively collected. The other more important reason is that there's an immune component to cancer, right? When cancer is developing, that elicits an immune response. So looking at patterns of methylation in blood might help us provide a more holistic understanding of the development of that disease. Now, what a lot of these early studies, essentially all of these early studies showed, so I'm just giving you some citations here for studies of DNA methylation in different types of cancer. What all of these studies showed is that there were marked differences in DNA methylation between cancer cases and people that didn't have cancer when we measured methylation in blood. However, the question that all of these studies were left with was, what is actually driving these associations? Okay? To begin to answer this question, there are a few key features of DNA methylation and the way in which it's assessed that need to be considered. Okay. The first is the fact that DNA methylation is tissue specific. So that is, different tissue types throughout the human body have a distinct methylation signature. And that shouldn't be surprising, as DNA methylation is part of what gives rise to the phenotype or identity of different tissues. So this is a figure from a paper that was published by a colleague of mine, Brock Christensen. And this shows this idea of tissue specificity of methylation nicely. So here, the columns are different samples, and they're color-coded according to what anatomical region throughout the human body that sample was actually taken from. The rows, and they're actually difficult to see because there's so many of them, there's tens of thousands of rows here. Each row represents a different CpG in the genome, and the elements of this heat map reflect the methylation level of a certain CpG for a certain sample. And remember, yellow's unmethylated, blue's methylated, black somewhere in the middle. So what we're depicting in this heat map is a hierarchical clustering of samples taken from different regions of the body based on their methylation signature. And we can see that different regions of the body cluster out fairly nicely. If we look on the right-hand side, these purple samples are all samples taken from the placenta. This group here are all blood samples, but they subcluster depending on where the blood was taken from. Was it taken from an adult, or was it taken from an infant? And then over here, we have a bunch of different solid tissue types. Okay? Take-home message, DNA methylation is tissue specific. Now what's also really interesting is these are all different blood samples, but we can drill in even finer, and we can look at different white blood cell types. So if we create a hierarchical clustering of methylation across different white blood cell types, we see the same type of pattern that emerges. That is, different cell types have their own unique DNA methylation signature. Okay? Monocytes, granulocytes, B cells, NK cells, T cells, these are all different white blood cell types, and we can see that their methylation signature differs across this set of CpGs. So, why is the cell specificity of DNA methylation potentially problematic for studies of DNA methylation in cancer when methylation is assessed in a heterogeneous tissue like whole blood? So, the fact that DNA methylation varies across different white blood cell types, together with the fact that we would expect the cell distribution or cell composition to be different between cancer cases and controls, because cancer elicits an immune response, means that we have that cell distribution has the potential to confound analyses of DNA methylation when measured in heterogeneous tissue type. Confounding, what we're talking about is, I don't know, a few years ago there was a study published looking at the relationship between coffee and lung cancer. And this study got a lot of press because it found that there was a statistically significant association between those that drank coffee and those that got lung cancer. Well, it turned out that there was this lurking variable called confounder, which was smoking. See, coffee drinkers were more likely to smoke, and we know that smoking is a risk factor for lung cancer, and that analysis failed to account for smoking in the comparison of coffee drinking and lung cancer. So that's what we term a confounder. So we have a confounder here, which is cell distribution. That can mess up our analyses, and we want to be able to correct for that. So let me give you an analogy to help make this a little bit more clear. Okay, imagine that I'm a fisherman, okay, and I've got two lakes. One of them's polluted and one of them's clean. And so I cast my net out into the lake and I pull in all of the fish, and what I do is I fillet all the fish that are in my net. And the meat that I get, I combine it all and I make my McFillet sandwich, okay? I have no disclosures here. I'm not being paid by McDonald's, although if there's anyone that works for McDonald's in the room that wants to pay me, I'd be happy to chat afterwards. So now let's suppose that we take these two fish fillets and we compare them between the polluted lake and the clean lake, and let's say they taste different, okay? Now there's three possibilities to explain why the fish fillets taste different between the polluted and the clean lake. The first is that the pollution is actually altering the way the fish taste. In other words, fish that are in the lake with pollution, it's like seeping into the pores of the fish, and it's actually changing the taste of the fish that way. The second possibility is that pollution is changing the distribution of the fish. See, evolutionarily, there may be certain fish that are better suited to live in the lake that has the pollution than other fish. And so we know that salmon tastes different than cod, so that might explain why the fish fillets taste different. And the third possibility is that it might be some combination of one and two. So we have the same type of thing with cell composition, right? Our polluted and our clean lake are like our cancer and our non-cancer patients. The distribution of fish are like the distribution of cell types. And the McFillet, fish fillet sandwiches are like the DNA methylation profile in whole blood or some mixed biospecimen. All right, so to begin to understand the extent to which variation in immune composition between cancer cases and controls might be influencing the results of some of those early studies that I was discussing that reported marked differences in methylation between cancer cases and controls in blood, we investigated whether CpGs that are differentially methylated or exhibit a different pattern of methylation between different white blood cell types, if they too allow us to separate or distinguish cancer cases from controls. If it turns out that this is the case, then that would suggest that some of the marked differences that were being detected in those early studies were merely being driven by the fact that the immune composition is different in people that have cancer and people that don't. That's not super interesting, right? We already know that that's the case. So we investigated this question using three publicly available data sets. And these three data sets spanned three different solid tumor types. One of the data sets was a case control study of head and neck squamous cell carcinoma. So there were 92 people that had this cancer and 92 controls that were cancer-free. An ovarian cancer data set that consisted of 266 ovarian cancer cases and 274 cancer-free controls, and a bladder cancer data set which had 223 cases and 237 controls. Now basically what we did was, now these are kind of the same heat maps that I was showing you before, except I'm flipping it, I'm transposing it. What we're doing here is we are finding the CpGs that exhibit a different methylation signature between different white blood cell types, and we're asking the question, are those same CpGs differentially methylated between people that have cancer and people that don't? So that's precisely what I'm depicting here. So each one of these points, the blue, the green, and the purple, represent the three different studies that I just described. So head and neck is green, blue is ovarian, and the purple is bladder. And what each one of these points represents is the minus log 10 p-value from a statistical test comparing the methylation signature of that CpG between cancer cases and controls. So let me provide some context here. The higher up on this plot that point is, the bigger the difference in methylation between cases and controls for that specific data set. So what we see here is out of the 50 most discriminative CpGs between the different cell types, we have 47 and 48 of them that achieve statistical significance in the head and neck and the ovarian cancer data set. And observing by random chance 47 and 48 out of 50 is incredibly unlikely. And in fact, one could run a statistical test on that and find that you've got basically a 1 in 100,000 chance of observing that. Now, the results are far less compelling for bladder But even observing 8 out of 50 CPGs as being statistically significant is still marginally significant in terms of its unexpected nature if one were to run a statistical test. Now, the results for bladder are also maybe not entirely unexpected, as nearly all of the bladder cancer cases that were in this study were receiving treatment with a chemical called BCG, which is a known immunotherapy. So that could certainly have an impact on the results. What I think is even more compelling is if you take these 50 CPGs that discriminate different white blood cell types, and you basically build a prediction model for predicting who has cancer and who doesn't, you wind up finding that for the head and neck, ovarian, and to a slightly lesser extent bladder, that a prediction model comprising these CPGs very accurately predicts who has cancer and who doesn't. So, what the results on the last 2 slides tell us is that the findings of early studies that were looking at DNA methylation in blood and comparing it between cancer cases and controls can in part be explained by differences in cell composition. So what are the ramifications of failing to adjust for cell composition when we conduct such analyses? So the first and clearest demonstration of this was published in 2012 by Liu in the journal Nature Biotechnology, but what I'm showing you here is some data from my own work. So in the top hand panel, the statistical model we're actually fitting to relate DNA methylation to case control status is given here. So Y is a variable that represents the methylation status for a CPG in the genome. X is an indicator of whether or not a given sample was a case or control. And so what I'm interested in actually statistically testing is whether or not this parameter alpha-1 is equal to 0 or not. So if I do an unadjusted analysis, and by unadjusted I mean I'm not accounting for cell composition when I actually fit the statistical model, we see that we have a huge number of CPGs that are being tested that are actually coming out as being statistically significantly different. And this volcano plot visualizes that. Again, minus log 10 Q value on the y-axis. The higher the point is on this plot, the bigger the difference in methylation is for that specific CPG between the cases and controls. Now what we see is when we adjust for cell composition, so we augment our statistical model with a term that accounts for the different proportions of cell types in the sample that we're profiling methylation in, we see that there's an attenuated effect. In other words, a lot of the things that were previously being declared as significantly different between case and controls go away. And the reason they're going away is because differences in immune composition is explaining those differences. And so when we adjust for immune composition, the effect goes away. All right, so what we have here is confounding due to differences in the white blood cell composition between study samples, people that have cancer and people that don't. So how do we address this from an analytical perspective? So the way that we could address this from an analytical perspective is the way that we address any other potential confounder when we're doing a statistical analysis. We obtain a precise and accurate measurement of that potential confounder, and then we adjust for them in our statistical models. So one solution involves doing what's called a complete blood cell count on the blood at the time of collection. So like when you go into the doctors and they take blood, part of what they do is a complete blood cell count. And that gives the fraction of lymphocytes and neutrophils and monocytes. This approach has some drawbacks though. First, it can't be used on stored blood. So when a doctor takes blood from you, they essentially immediately need to do the complete blood cell count on that sample. They can't go freeze it and then do the blood cell count. It just doesn't work that way. The other limitation of doing a complete blood cell count and adjusting for it in our analysis is that complete blood cell counts lack granularity. Lymphocytes is a very broad category of cell types. There are many sublayers to that. So we don't have a very granular approach here. The other thing we could do is what's called flow cytometry. Now flow cytometry is going to give us the granularity that complete blood cell counts can't. However, it also requires fresh samples. So we can't do flow cytometry on blood samples that have been sitting in a freezer, which is very, very common for epidemiological studies. The other thing about flow cytometry is it's an additional expense, right? I mean, we're talking about a couple $100 plus having to pay the technician who's actually doing it. So it's an added cost to the study. Can we be smart about this? So this is where math comes into play. Okay, we've got some math on this slide. A lot of biology up to this point, but now we're starting to talk about how math can be used to solve this. So remember back that one of the key features of DNA methylation is its tissue specificity. So what we can do is we can leverage this feature of DNA methylation as a means towards deconvoluting the cellular composition of a mixed biospecimen that we've profiled methylation on. So if we profile methylation in whole blood, that data is represented by Y. And Y is a weighted mixture of the methylation signature coming from the individual cell types that underlie that sample. So S1, S2 are the methylation signatures for, say, lymphocytes, monocytes, and granulocytes. And these Ws reflect the proportion of each one of those cell types in the mixture. So they're contributing to Y proportionally depending on how much of the mixture they make up. Okay? So written in matrix notation, we have this equation here where E is just a random error term. Now, we know what S is, right? We have actually gone out and profiled the methylation signature of purified isolated cell types. In a given study, Y is DNA methylation profiled over whole blood, so we have that as well. So the only unknown in this equation is W, okay? So the problem involves finding the W that minimizes the squared error loss function, subject to the constraints that the W's across the number of components in the mixture, which is represented by K, such that they sum to 1, and that makes sense. The proportions of the individual cell types should sum to 1, and that they should be proper proportions, and that they're bounded below by 0 and above by 1. So this is actually a simple mathematical problem, and it can be addressed using a technique called constrained projection or quadratic programming. So while constrained projection or quadratic programming is a standard statistical technique, its application for the deconvolution of cell composition using DNA methylation was first described in 2012 in this landmark paper that was first authored by Andy Hausman, who happened to be my advisor when I was a PhD student. This method has since been dubbed the Hausman method. That's when you know you've made it as a statistician or a mathematician. When you've got like a method named after you, you're good to go. So the idea is actually really simple. Step one is we need to identify the fingerprint of each one of the cell types. So what is the methylation fingerprint of each of the cell types in my sample? And once I've done that, all I then need to do is find a W that minimizes the squared error loss function subject to these constraints. A very simple mathematical problem. Okay? I'm going to skip over this for a moment. So, how does this technique actually work? So, in the original paper describing this technique, it was applied to some experimental data, and it achieved really good results. But those were experimental data. How does it perform in real humans? So to answer that question, we utilized a publicly available data set that was published in 2012. And this data set consisted of DNA methylation profiles of blood for 94 subjects. And in addition to DNA methylation profiles in blood, they also had CBC measurements. So I have a gold standard measurement for cell composition. So what I can do is I can apply this technique to the data, predict cell composition for the samples, and then compare my predictions to the CBC measurements to see how close am I to the actual truth. So on average, there was really good consistency. Across these 94 samples, monocytes were predicted to be 17.9%, or that was the CBC measurement, and using the Hausman method, we predicted 17.6%, pretty darn close. For lymphocytes and CBC, 82.1% versus 81.3% predicted by the Hausman method. Now, these plots right here depict the predicted cell proportion for monocytes and lymphocytes as a function of their CBC measurements. So while on average we're doing really well, on an individual level, there's a lot of room for improvement. Ideally, what we would like to see, if we've got a perfect prediction tool, then all of these points would lie along the line of unity. We can see that that's far from the case. There's a lot of deviation from this line, thus a lot of room for improvement. So that's where I came into play and started thinking about how can we actually improve this technique. The mathematical technique is pretty solid, so I'm going to leave that aside. So if I'm going to improve this technique, the way in which I would do that is by better selecting the fingerprint, the DNA methylation fingerprint for each one of the cell types. So that's where the gains can really be made. So let me kind of give you an example. There are two heat maps on the slide. I want to focus your attention on the heat map on the left-hand side. So in the original Hausman paper that was published in 2012, they proposed this approach that was based on ANOVA ranking. It's not really important what that is, but it was based on ANOVA ranking for selecting the library that was used to deconvolute cell composition. Now, what we can see from this library is that some of the cell types are well discriminated from others. Like granulocytes and monocytes, we've got a chunk of CPGs right here that are very different. So they're well discriminated. B cells are well discriminated from monocytes and granulocytes, as are CD8s and B cells. However, look at CD8s, CD4s, and NKs. Across this entire panel of CPGs, they exhibit a very similar methylation signature. That's problematic in that the algorithm can't determine which is a CD4 from a CD8 or a CD4 from an NK. We lack the signal to discriminate them. A better strategy for selecting this library would be to take the CPGs and take one cell type at a time and compare it to the remaining cell types and rank order the CPGs that best discriminate that cell type from all of the remaining cell types. And if we do that, we end up with a heat map like this. Now, there's still room for improvement, but we can see that now at least we have some signal for discriminating NKs from CD8s and CD8s from CD4s. So we're doing a little bit better. Now, what does this actually mean in terms of prediction performance? So the two different approaches we were just looking at, the first one was called the ANOVA approach, the second one was called estimate cell counts. And I've got two different metrics of prediction performance. The R squared is the proportion of variance in the true proportions that's explained by our prediction. So the R squared is bounded below by 0 and above by 1. And we want that number to be as close to 1 as possible. If we've got a perfect prediction tool, the R squared would be 1. The RMSE is the average deviation of our predictions from the truth. So we want the RMSE to be low, as close to 0 as possible. So what we can see is that the second approach, with the exception of CD4 cell, always achieves a better R squared and always achieves a smaller RMSE. That being said, this approach still has some room for improvement. Now, we want these numbers to be as close to 1, and the NK cells 0.7, that's okay. 0.29 leaves a ton of room for improvement. So is there a way that we can even further improve on this approach? And to that, I present you with another analogy. So we're going to harken back here to the early 90s when the Bills were legit playoff contender. But look, they're getting better. This is the year, all right? This is the year. So imagine that we want to select the best 11 players to put on the field at any given time from a 53-person roster. Now let's forget about special teams, offense, and defense. Okay, we've got 53 men on a roster, and we want to select the best 11 players to put on the field. So if we were to evaluate the performance of every single combination of 11 players out of 53 to find that optimal set, there would be 762 billion combinations. Clearly a very challenging computational problem. This problem is exponentially worse in the context of the methylation data we're dealing with. To give you an example, let's pretend that there were 1,000 really good discriminatory CPGs between the cell types, and the optimal library consisted of 100. If we were to evaluate every combination of 100 from 1,000, that would be 6.4 times 10 to the 139 combinations, and that's more than the number of particles in the universe. Can't do it. So how can we get to that optimal library faster? So let's use this as an example. The way that we might do this is, let's say that we begin by randomly selecting 11 players from the roster of 53, complete random selection. And let's say that we evaluate the performance. We have some metric for evaluating the performance of those 11 players, and we write that down. And now what we do is we go through those 11 players and we remove one at a time, and we evaluate the performance. We then compare the performance of that one player that was taken out to the performance of the full squad. Now, if the team performance improves without Jim Kelly being in the lineup, clearly very unlikely, then we'd want to downweight the selection that Jim Kelly has selected in subsequent iterations of this algorithm. If the team performance without Jim Kelly does worse, which is more likely, then we want to upweight the selection that Jim Kelly has selected in subsequent iterations. And if the team performance is equal with or without Jim Kelly in the lineup, then his selection probability should remain the same in subsequent iterations. Now we go through and we repeat that process for each one of those 11 players. And what we've done at the end of the day is we've now updated their selection probabilities. And we go back to the original. We now randomly select a new 11, but now selection probabilities are changed somewhat. The idea is that by repeating this process numerous times, that a couple hundred iterations into the future, that we actually converge on the optimal solution. So rather than having to go through 762 billion combinations, we get to that optimal solution a lot quicker. So can we adapt this strategy for selecting CPGs to build this library that we're using for cell mixture deconvolution? And the answer to that question is yes, but we need to define a few things. So first of all, we need to formally define how we assess prediction performance. Well, that's fine. We can use the R-squared and RMSE that I already talked about. Those are good performance metrics. The other question that we need to grapple with is, how do we assess the contribution of specific CPGs as they relate to prediction performance? Similar to kind of removing one player at a time. Thirdly, we need to formalize how we update the selection probabilities of each CPG at each iteration of the algorithm. So I do have some of the details here for those that are interested. I don't want to get in, too deep into the weeds, but all of this information, all of these formalities are described in a paper that we published in BMC Bioinformatics in 2016. And so this is just a description of the algorithm. For anyone that's interested, I'm happy to sit down with you afterwards and kind of go through this algorithm. I do want to show you this. So this is essentially what's happening. This is a matrix. Now, the selection probabilities of all the CPGs, so all of the CPGs are in this matrix, and they all have an equal probability of being selected at the first iteration. Now, what happens is as we move through this algorithm, some of the CPGs were deemed to be not helpful to prediction performance, and thus their probability of selection went down. So those would be like CPGs that are red. Some of the CPGs are being found to be helpful to prediction performance, and thus we are updating their selection probability. And the idea is that we're converging on a better library as we move through iterations. And this can actually be seen if we plot out the performance metrics I just described as a function of the iteration number. So the RMSE, remember, we want that to be low. And as we move through the iterations of this algorithm, the RMSE gets lower and lower and lower and lower and lower until we converge on an optimal library. The same thing with the R-squared. That goes up and up and up and up until we basically asymptote near 1. Okay, so this is all fine and good. It seems to be interesting in theory, but let's actually put this to the test. So the biologists that I work with conducted this experiment where we had one data set that was created in which to train this algorithm, to basically build this library. We had a separate data set that they had created where what we did was we purchased cells from a company. We then mixed the cells in predetermined proportions that I gave to my investigators. So we know the ground truth of the cell composition, the proportions of each one of these cell types, and then we profiled DNA methylation over that mixture, so we actually had a gold standard ground truth to compare our predictions to. And here's what the results look like. So along the y-axis of each one of these plots, so each of these plots is a different cell type. The y-axis is our predicted fraction, the x-axis is the observed fraction. And again, the best prediction model that we could have would have all the points lying along the line of unity. Okay, that's not the case. We're not all along the unity. there's some variability here, but we're pretty darn close. And if we look at our metrics of R squared and RMSE, the R squared is close to 1 in all of the different cell types, and the RMSE is pretty low for all of these cell types as well. So the algorithm seems to be working quite well. We have since validated this algorithm using an entirely different set of samples and an entirely different array technology for assessing DNA methylation, and have also seen similarly good performance. Again, for each one of these cell types, our predictions are very close to the truth. We see these points lying aligned or very close to the line of unity. Okay, so in the last 5 to 10 minutes, I want to talk about some recent work where we're actually applying these methodologies to some real cancer data sets, and that's where I kind of want to leave you. So, the utility of these cell composition estimates, they're basically 2 broad categories. The whole impetus for predicting cell composition stemmed from studies where we've got cancer cases and controls, we want to identify differences in DNA methylation, but we have this confounding due to cell composition. So what we've provided is a technique for estimating cell composition. Once we have those estimates, we can control for them in our statistical models and reduce the effect of confounding. Okay, so that's one way we can use these estimates. The second way is that the estimates themselves may actually be of interest. We can look at these estimates and ask questions about, are changes in cell composition, are they associated with risk of cancer? Or if you already have cancer, are they associated with your prognosis? Because if you think about it, that would be a really valuable piece of information for a clinician who's treating a cancer patient. If they look at their cell distribution and they see that the neutrophils are really high, they might want to put that person on a more aggressive treatment because high neutrophils is associated with poor prognosis. So it can be used for individualized therapy between the clinician and their patients. So I want to focus now in the next 10 minutes on some studies I've been involved in where we actually use these cell estimates to understand cancer risk and progression. So if you've gotten a complete blood cell count at the doctor's, as I mentioned, it measures lymphocytes, monocytes, and neutrophils. There's one marker that can be derived as a ratio of 2 of those, and that's called the neutrophil to lymphocyte ratio. And it is exactly the way that it sounds. It is the ratio of neutrophils to lymphocytes. There have been a remarkably large number of studies that have shown that elevated NLR is associated with increased cancer risk, increased risk of cardiovascular disease, increased risk of type 2 diabetes. There have also been a huge number of studies that have shown that if you have one of those diseases, that elevated NLR is associated with poor prognosis. So, given that our technique allows us to estimate the components of the NLR, we can create what we term the methylation-derived NLR. And that's simply the predicted fraction of neutrophils or granulocytes divided by the predicted fraction of lymphocytes. We can leverage DNA methylation to get those predictions, thus we can derive an estimate of the NLR. So, remember those 3 data sets that we talked about earlier in this presentation? Well, we also have survival information on each of the cancer cases in those data sets. So those cases have been followed up in some cases for as much as 10 years. And so we know who died and who is still alive after 10 years. So we can take the methylation-derived NLR that I just described, and we can ask the question, is it associated with cancer survival? And across each one of these cancer data sets, this is a brain cancer data set, bladder, head and neck squamous cell carcinoma, we see that elevated NLR is associated with poor survival prognosis. So these plots are called Kaplan-Meier survival plots, and the lower curve is the curve for people that had a high MDNLR. And basically what this means is that when you have a high MDNLR, you are dying more frequently and quicker than if your NLR is lower. So this is potentially a potentially useful prognostic marker. So the reviewers of our paper argued that, well, you might have confounding in these studies. All right, so we can fit statistical models where in each one of these studies we adjust for potential confounders. Like age is a good predictor of survival, so we'd want to adjust for that. We can adjust for sex, smoking status, tumor stage at the time that they presented. And nevertheless, in all of these instances, we see that elevated NLR is associated with poor prognosis. So that association is robust to adjustment for potential confounders. We also see that this MDNLR measure is associated with cancer risk, so the chance of actually getting cancer if you're cancer-free. So in the head and neck squamous cell cancer data set, what I'm showing here is the distribution of the MDNLR in controls. These are people that don't have cancer, and in cases. And we can see that the case distribution, though there is a decent amount of overlap, it's shifted towards the right, suggesting that cancer cases have higher MDNLR as compared to cancer-free controls. The other interesting thing is that this MDNLR can actually be used to very highly accurately predict who has cancer and who doesn't, as evidenced by this ROC curve and the corresponding area under the curve. We see the same type of thing in ovarian cancer. What I think is really interesting here is this curve is the distribution of MDNLR among pretreatment cases, so cases before they actually got treated with chemotherapy and radiation. This is the curve in post-treatment cases, and this is the curve in controls. What's really interesting here is that the treatment group is starting to revert back to a distribution that is reminiscent of the controls that do not have cancer. So that would suggest that, globally speaking, that the treatment that these people were being put on is actually effective. It's working. It's moving that MDNLR back to what it looks like in cancer-free individuals. So the last study that I'll discuss with you is kind of recent work with some collaborators of mine from the University of Utah and Emory University. I should mention that these two studies were what we call case control studies. That is, the biological samples were taken after the people already had cancer. So reverse causality is a possibility. What that means is, was the disrupted MDNLR there before they had cancer, or did cancer lead to the disrupted MDNLR? To really gain insight into that question, we actually have to have we have to run a prospective study and have the samples collected before the actual disease develops to see if it's truly a risk predictor. And so that's what we've done in this study. This is a study of lung cancer, and it's based on a massive study called CARROT. And there were 14,000 heavy smokers that were enrolled in this CARROT study. They were followed for 15 years. Now, some of these people got cancer, and some of these people didn't. I should also mention that at the time of enrollment in the study, blood was collected from every single one of these individuals. So we follow them for a certain period of time. Some get cancer, some do not. What we want to see is there a pattern of this MDNLR in blood taken well before they got cancer that predicts who goes on to get cancer versus who doesn't? And this is a very busy table, but I just want to draw your attention to the top. What we actually saw was these samples were taken on average 4.5 years before formal diagnosis was made. And what we saw was that the people that eventually went on to get cancer did exhibit an elevated MDNLR, which suggests that there are things that are happening in the immune system before cancer is even detectable that might be a precursor to the development. So that again gets to the clinical utility of this and why this could be potentially very valuable information for a clinician who wants to basically identify people that are a high risk for disease. There are a number of other ongoing developments in this area. What I've talked about today is deconvoluting mixed biospecimens that were taken from adults. But some of my colleagues are working on these deconvolution algorithms for different types of biospecimens. So umbilical cord blood. So if we're studying infants and diseases that are, show up shortly after birth, we may want to look at DNA methylation in cord blood that's taken at the time of birth. And so we need to worry about confounding by cell composition in those studies as well. And some of my colleagues are working on deconvolution algorithms for those studies. I also have some colleagues that are working on extended libraries. What that means is our library only allows you to predict 6 different cell types. Though they are the major components of blood, there are many, many different cell types in blood, and we are in the process of extending that library to enable predictions of additional cell types. I should also mention that no talk on deconvolution is complete without at least mentioning approaches that are termed reference-free. These reference-free approaches don't, unlike our approach, which you need the individual signal for each one of the cell types in order to enable deconvolution. These approaches do not depend on the individual signal and are pretty similar to the blind source separation techniques that are used in the audio and image literature. All right, so take-home messages. So blood-derived DNA methylation presents unique challenges. There's this issue of confounding by cell heterogeneity, and even if cell heterogeneity isn't a confounder, it's still an important source of variation and should be something that's accounted for in our statistical method. Differences in cell composition is a major factor that explains the results of some of these early studies that were looking at blood-based DNA methylation in cancer. And the cell specificity of methylation can be leveraged for deconvoluting cell composition. So that is a combination. I think that this work, what really excites me about it is that it truly is team science. Like, as a mathematician or a statistician only, this work would not be able to happen. I needed a team of biologists, we needed a team of mathematicians and statisticians to make this work possible and to communicate in a language that we could all understand. So with that being said, I'd like to acknowledge some of the people that have been instrumental in this work, as well as the funding that supported this work. So up on the top left-hand side, this is the lab that my colleague and I run at the University of Kansas Medical Center, and these are some of the students that we have the great privilege of working with that are really the boots on the ground people that are doing this work. I can get up here and talk about it, but it's the students that are really putting in the time and effort out of class. I'd also like to
Undergrads or grads, sorry.
All graduate students, all graduate. Yeah, at the medical center, there's no undergrads, all grad. I'd also like to thank the collaborators, my mentors, my friends that have been there from when I was a graduate student all the way through my career as a faculty member. Carl Kelsey at Brown University, John Winke at University of California, San Francisco, Andy Hausman, who's since retired from Oregon State and was formerly at Brown, Brock Christensen at Dartmouth College, and his postdoc, Lucas Salas, who is at Dartmouth as a postdoc and will be staying on there as a faculty member. I also like to thank the sources of funding, the NCI, the NIH, and KAMBRI. So at that stage of the game, I've hit you hard enough. I'm happy to take any questions. Thank you.
Any questions for